Good afternoon, professors. Thank you for taking time to hear about my thesis. My name is Maria Arian Agatha Victoria David, and today I will be talking about my thesis, the prediction of a novel epitope-based peptide vaccine against Plasmodium falciparum using an immunoinformatic approach. Malaria has been a disease that has been plaguing us for centuries. It causes serious and even fatal complications on humans. Its primary mode of transmission is by mosquitoes from the genus Anopheles. The bite of an infected mosquito transmits a parasite that enters the victim's blood system and travels into the person's liver, where the parasite reproduces. The CSP, or the circumspermatozoite surface protein, are proteins that can be found on the surface of the spermatozoite. Some of its main functions are motility, attachment, and entry of the parasite into hepatocytes or liver cells. And then in 2019, nearly half of the world's population was at risk of malaria. And most of malaria cases and deaths actually occur in sub-Saharan Africa. However, the World Health Organization regions of Southeast Asia, Eastern Mediterranean, Western Pacific, and the Americas are also at risk. According to the latest World Malaria Report, there were 229 million cases of malaria in 2019, compared to 228 million cases in 2018. The estimated number of malaria deaths stood at 409,000 in 2019, compared with 411,000 deaths in 2018. So currently, treatment for malaria is very expensive, and a fully effective vaccine is still unavailable. The leading vaccine is called Mosquirix, so it's by GSK, and as of 2020, it only has 56% efficacy, which also decreases every year for 4 years. Currently, the vaccine has moved on to phase 4 trials, but there are still challenges on the vaccine itself. One of the reports about Mosquirix has speculated that CSP-based vaccines that eliminate or reduce responses to variant epitopes could improve the overall efficacy of Mosquirix. So this study performed computational analysis of immunological data by predicting multiple epitopes for a vaccine against malaria by utilizing the circumspermatozoite surface protein from the parasite Plasmodium falciparum. Using immunoinformatics will reduce the time frame and the cost of vaccine development for malaria compared to using conventional approaches. The objectives of the study are: first, we want to identify immunogenic and antigenic CTLs, HTLs, and LDLs that can potentially be used for improving the vaccine efficacy. And also, we want to predict, simulate, and evaluate possible immune responses to the predicted peptide vaccine. So by selecting or eliminating unnecessary epitopes, the resulting epitopes can be of help in improving the overall efficacy of CSP-based vaccines, such as Mosquirix. Doing this, we also reduce the time frame and the cost of vaccine development and maybe even supplement existing information about CSP. So, how was this done? First, the CSP sequence was obtained from a malaria endemic area, which is the Tak province in Thailand, from the years 1996 to 1997 and 2006 to 2007. So this sequence was obtained from the NCBI database, protein database, and then screened it for CTL, HTL, and LBL epitopes using the NetCTL, IEDB, and IBCEL respectively. CTL selected epitopes were then tested for their ability to induce an immunogenic response. Using IEDB class 1 immunogenicity tool, their ability to induce an immunogenic response was tested. Only epitopes that showed a positive value for immunogenicity and antigenicity were kept for the next stage of evaluation. And the remaining candidates were assessed for their toxicity and allergenicity using toxin-pred and allertop, respectively. And the MHD1 alleles were predicted with IADB. The HDLs, selected epitopes, were subjected to the assessment of their antigenicity using Vaxogen and were thereafter assessed for their toxicity and allergenicity. The cytokine inducibility was also tested for their ability to induce interleukin 10, interleukin 4, and IFN gamma. Then the selected CTL, HTL, and LBL epitopes were subjected to mutation screening, where randomly selected circumferentozoid surface proteins were taken from the NCBI database and aligned using the Crystal Omega server. The results were viewed using MView multiple alignment viewer, and after that, the population coverage analysis was also done for the CTL and HTL epitopes. And then after which the selected CTL, selected HTL, and selected LBL epitopes were added to the peptide vaccine construct, where we also had the linkers and the adjuvants. The resulting vaccine construct was then subjected to antigenicity, toxicity, and allergenicity prediction. It was also subjected to assessment of physical chemical properties, and finally, in silico immune simulation was initiated, where a 4-week minimum interval between the first, second, and third dose were used. So these are my selected epitopes. These epitopes are all immunogenic. They're all antigenic, non-toxic, and non-allergenic. These are also the same epitopes that were used to make the vaccine construct. In the vaccine construct, we can see that 3 types of linkers were added, and the adjuvant used were the 50S ribosomal protein L10. This adjuvant has a functional role as a TLR4 or toll-like receptor 4 agonist, which basically helps in stimulating dendritic cells, phagocytes, and other immune responses. So, and then EAK was the linker that was used to provide rigidity, and also to reduce possible interference of other protein regions in the interaction between the adjuvant and the receptor. The remaining linkers were used mainly considering their ability to induce immune responses. So the combination of my CTL and HTL epitopes had a high population coverage in our target areas, such as in various regions of Asia and Africa. However, there are also regions such as in Central America where the population coverage can be considered low. And this could be attributed to 2 possible causes. First is that the prevalent causative in there is the Plasmodium vivax, which is another form of malaria. And second is that because there are certain alleles, HLA alleles, that are not as common in those places. However, doing the world population coverage, we get a we found that 99.96% could be covered by the vaccine construct, using the Clustal Omega multiple sequence alignment tool, 54 randomly generated sequences of the selected protein were processed and viewed using the MView multiple alignment viewer. Most of the epitopes were found to be present in the multiple sequence alignment. This supports our notion that the protein sequence chosen is highly conserved. This is also expected because the chosen protein sequence, according to studies, were found to be favored by natural selection and were found to have patterns that are consistent with balancing selection driven by interactions with the immune system of the vertebrate host. So the resulting vaccine construct was subjected to property testing using falciparum. The vaccine construct was found to have a particularly high antigenicity with Vaxogen. This could be because in Vaxogen there is an option that you can choose a target organism. The construct is also found to be non-allergenic and non-toxic, which means that it should be safe. The instability index, which is a measure of the stability of a protein, uses a reference value of less than 40. In this case, the vaccine construct is less than 40, which is 31.85, and so we can consider it stable. The aliphatic index plays a role also in protein thermal stability, and with a high aliphatic index, proteins are more likely thermally stable. In here, we have a very high aliphatic index, which suggests that our construct is thermally stable, which is a good sign as malaria is prevalent in tropical countries. And lastly, the Gravy value, or the grand average of hydrophaticity index, is negative 0.674, which means that the vaccine is possibly hydrophilic, which could increase humoral immune response. So Here are the immune simulations. We can see here in graph A that it's the immunoglobulins and the immunocomplexes. We can see here that in Plasmodium falciparum, IgG1 and IgG1 plus IgG2 and IgM responses to the parasites are incremented during protection to the disease. The primary response was characterized by the rising levels of IgM, and the secondary and tertiary responses saw a marked increase in the B cell populations in levels of IgG1 plus IgG2, IgM, and IgG plus IgM antibodies with a corresponding decrease in antigen concentration. This profile indicates development of immune memory and consequently increased clearance of the antigen upon subsequent exposures. A similarly high response was seen in the helper T cells and cytotoxic T cells cell populations with corresponding memory development. And here, lastly, in this figure, levels of IFN gamma and interleukin-2 slightly decreased following repeated exposures to the antigen. Although the cytokine response slightly decreased, their results still indicate high levels of IFN gamma. Thus, we can expect that cytokines can still be produced, and thus we can also expect a humoral response. So the recognition of CD8 plus T cells is actually important in the destruction of intrahepatic parasites like Plasmodium falciparum. And once IFN gamma is produced by CD4 plus and CD8 plus T cells, protective immune responses are expected to follow. The danger signal, which is D here, which is the blue line, it's actually just basically alarm signals that are released when there are infected cells, just to let the immune system know that there is a problem requiring an immune response. So the combination of epitopes with high scores of antigenicity and immunogenicity has been of great help in increasing the antigenicity of the vaccine sequence, alongside the use of highly effective adjuvant and linkers. In this study, CTL, HTL, and LDL epitopes were predicted and assessed according to various properties such as allergenicity and toxicity. But most importantly, their antigenicity and immunogenicity were assessed. These two factors have the most brutal aspect in any part of vaccine development. The designed vaccine construct was found to be antigenic and immunogenic, with physical properties that are admirable as a complementary tool in malaria vaccine development. So as a recommendation, because the study was performed using immunoinformatics, it could serve as a basis in the development of an actual peptide vaccine. However, before all of that, we must be certain that all of the properties and check out. And it is suggested to perform further testing evaluations such as molecular docking and structure prediction to find possible binding sites for the antigenic molecules and to ensure the safety and plausibility of the vaccine construct before proceeding to in vivo or in vitro studies. And that's all. Thank you for listening to me.